gene drive organisms, to at least some extent, and some gene drive organisms to a large extent, mirror pathogens in the sense that even when they're being experimented with in the lab, they have the possibility of escape, and when they escape, in many circumstances, they may have the ability to spread. And this is precisely what you attempt to control against when you're trying to do pathogen biosafety, and you adjust the level of safety that you use in labs according to the threat. And it seems to me that gene drive organisms fall into a very similar category. A traditional genetically modified organism may pose a variety of biosafety dangers. It is typically not designed to suppress other variants and to assert itself over the entire or a very large percentage of the wild population. Um, so I guess in my experience, when I think about how, rather than just a more traditional organism, how would you go about trying to adequately regulate the laboratory use of a gene drive organism, which has that capacity to potentially spread from one organism to another and to move through the environment and to physically move from place to place. It seems to me that the most appropriate analogy in the world of biosafety is that of pathogens. Um, that's an idea that some scientists don't like, but I think that when you stack it up and you look at the potential effects of a accidental release of a gene drive organism that's designed to spread itself, and which is uh, by definition not thoroughly tested if you're just creating it in the lab. I think that's the appropriate type of regulatory regime to look towards.